The aging process is often portrayed as an inevitable unraveling of biological order, a decline pre-programmed into our very genes. This perspective lends itself to the popular notion that aging is a disease, something to be targeted, halted, or reversed through pharmaceutical means. Yet, a closer look at mitochondrial biology reveals a different and perhaps more empowering view that the so-called mitochondrial aging is not the result of a mysterious internal timer, but rather the visible result of chronic nutrient deficiency. Mitochondria, the energy generators of our cells, do not simply break down with age. They wear out from decades of inadequate biochemical support. When we view mitochondrial decline not as an unavoidable pathology, but as a starvation of essential cofactors, aging becomes a nutritional problem, not a genetic curse. Mitochondria require a suite of nutrients to function properly, each one a cog in a finely tuned metabolic machine. Coenzyme Q10 B vitamins, especially B1, B2, B3, B5, and B6, lipoic acid, magnesium, and carnitine are just a few of the essential substances needed for optimal mitochondrial performance. These cofactors enable the electron transport chain to move electrons efficiently, generate ADP, and prevent harmful byproducts like reactive oxygen species, ROS, from accumulating. But when any of these nutrients are in short supply, either due to poor diet, metabolic stress, environmental toxins, or age-related absorption issues, the entire system falters. ADP output declines. Oxidative damage increases. Cellular stress mounts. What results is not just fatigue or weakness, but a progressive dysfunction at the core of all energy-requiring processes in the body. This energy bottleneck is central to the symptoms we associate with aging: loss of muscular strength, slowed cognition, poor recovery, and systemic fatigue. But these symptoms are not inevitable, they are biochemical consequences. If mitochondria are underpowered due to decades of micronutrient shortage, then the real culprit is malnutrition, not disease. This insight radically shifts our approach. Instead of trying to pharmacologically override mitochondrial decline, we could work to restore the cellular environment needed for mitochondria to thrive. Nutrient repletion through diet, lifestyle, and intelligent supplementation offers a gentler, more biologically compatible path to longevity. Moreover, the evidence supports this. Older adults with higher intakes of coenzyme Q10 and B vitamins tend to have better mitochondrial function, improved endurance, and sharper mental faculties. Clinical studies have shown that supplemental lipoic acid can reduce oxidative stress in aging tissues. Carnitine has been shown to improve mitochondrial transport of fatty acids and reverse some signs of frailty. These interventions are not miracle cures, they're nutritional restorations. They do not aim to conquer aging, but to nourish the systems that keep us alive and functioning. When mitochondria fail, it is not because they were designed to break. It is because their nutrient-dependent machinery has been starved for too long. The modern diet, high in calories but low in micronutrient density, offers short-term energy but little long-term support for mitochondrial resilience. Over time, this mismatch between caloric load and micronutrient availability creates internal energy debt. Mitochondria shrink, fragment, or even initiate programmed cell death. Organs begin to fail not because of chronological time, but because their energy factories have gone offline from neglect. From this angle, mitochondrial aging is not a mystery but a message. It is the body signaling that it lacks the tools to sustain high-output living. It is not disease in the strictest sense. There is no foreign invader or a runaway mutation, but rather a quiet collapse of metabolic infrastructure due to chronic undernourishment. This is a more optimistic vision of aging. It says we are not helpless against decline. We have tools, nutrients, habits, awareness that can nourish our mitochondria, restore our energy, and extend our health span. Rather than fighting aging as a disease, we might do better to listen to what aging is telling us: that we have unmet needs, that our cells are underpowered, not defective, that the key to vitality is not in blocking aging pathways, but in supplying the raw materials of cellular life. By reframing mitochondrial decline as nutrient starvation rather than dysfunction, we can abandon the notion that aging is something alien or pathological. Instead, we can see it for what it is: the cost of long-term biochemical debt. And if it's a debt, that means it can be repaid.